Yes, I'm going to be talking very briefly about a few different types of innovative early phase trial design. So there's going to be quite a big focus in terms of exactly what I'm saying on cancer trials, in particular drug trials. But I should just start by clarifying that a lot of this is really going to be relevant to other disease areas and other types of intervention as well. So I'm going to be beginning by going through some phase one designs. So I'm going to contrast what's known as the 3+3 design against a newer, more innovative method called the continual reassessment method. And then I'm going to run through what's known as curtailment in single arm trials and the potential advantages of that. Before I'll then discuss a variety of methods for improving on non-randomized trials, because non-randomized trials are quite common in phase 2 cancer settings, and there's been quite a lot of work done recently on how we can try and improve on the evidence that they provide. And as I go along, I'm going to be trying to note throughout what the advantages and the disadvantages are of these different approaches, but please do ask questions if anything isn't clear. Okay, so to begin with phase one trials then. So what is a phase one trial? Well, typically it represents the first experiment that you do in human subjects for your new treatment of interest. And your overall aim really is to identify what the highest dose is subject to certain toxicity constraints that you'll then move forward with in phase 2 trials. And this is usually referred to as the maximum tolerated dose, or MTD for short. And this is really based on the assumption that the efficacy of a treatment will increase with dose. So what are the key aspects then for these designs that you use in phase 2? Well, you need a toxicity outcome that's actually going to evaluate safety of your treatment. And typically this is a binary measure, so a dose limiting toxicity, or DLT for short, is very common in cancer trials in phase 1. You then are always going to need to specify a target toxicity level, so that's how much toxicity you're actually willing to accept on average in patients. So this will be what you're targeting at the actual MTD that you'd like to identify. And then you need a selection of doses that you're going to experiment with, including which particular one you're going to give to the first patient. And so choosing the range of doses is obviously quite an important thing, which this image I took from this paper by Grayling and colleagues tries to demonstrate. So obviously, if you pick your doses too low, for example, and the MTD happens to be above that, you're not actually going to be able to identify that. So always important to make sure you have a good range of doses. And then there are various other design aspects that you need to choose as well. So for example, you need to say what the maximum sample size is that you're willing to continue to, any stopping rules in terms of overall safety. So, if you're seeing far too many adverse events, when are we going to curtail the whole trial? And then typically for safety reasons, these studies are done very sequentially in very small cohorts. So you need to choose a size for these cohorts, with 3 being a typical amount. So then the final thing then that you need, a really crucial aspect, is your actual dose escalation design. So the design that's going to tell you how do you move between the different doses. And broadly speaking, there's 2 different types of designs. So there's rule-based designs and model-based designs. And I'm just going to contrast 2 of these, which are very well known against each other. So the rule-based design being the 3+3 and the model-based being the CRM, or continual reassessment method. So what is the 3+3 then? Well, this is a very commonly used design because of its simplicity. So essentially all you do is choose a set of doses you're going to experiment on, and you use the lowest dose as your starting dose. And then at your current dose, you recruit 3 patients into your trial. And if none of them experience a DLT, then you'll escalate to the next dose. Whereas if 1 out of 3 suffer a DLT, then you'd repeat and treat another 3 patients at the same dose. And if 2 out of 3 or 3 out of 3 experienced a DLT, then you de-escalate to the lower dose level. Then if you're at a point where 3 patients have already been recruited to your current dosing level and you recruit a further 3 patients, at that point, if 1 out of 6 have suffered a DLT or 0 out of 6, you'd escalate to the next dose level. If 2 out of 6 had suffered a DLT, you'd actually recommend that dose as the MTD. And if more than 2 out of 6 had experienced a DLT, then you de-escalate. And then you essentially just repeat steps 2 and 3 until you end up recommending an MTD. So this is really very, very simple. As I said, these are called rule-based designs because this is just a rule that you follow. It tells you exactly what steps you take when. So obviously, that's the fundamental advantage here. It's very, very easy to implement. It also has the advantage that there's no actual underlying statistical assumptions here, really. So whenever you make any statistical assumptions, you can run into trouble if those assumptions are violated. So it might be viewed as a good thing that this is so simple because it's not actually making any assumptions that you can violate. However, because this design has been used so much, it has been studied an awful lot now, and there really are quite a large number of disadvantages. So in particular, it's been shown that many patients actually end up being treated at subtherapeutic levels. Probably the biggest disadvantage, in my opinion, is this second bullet point here, that only your information from the current dose level is actually being used to make your decisions. But actually going about and figuring out which one is the MTD or what dose to give next isn't done very reliably. Another key thing to consider here is that there's no actual formal inclusion of this target toxicity level into the design. The 3+3 is basically based on the idea that around about a 33% target toxicity level is common in cancer trials. But that value actually isn't taken into account anywhere in the design. And then I've also included the fact that there's no underlying statistical model as a disadvantage as well as an advantage, because although you can run into trouble if you make assumptions and they are violated, quite often there are good reasons for making those assumptions, and you can make assumptions that will often hold and provide quite significant statistical advantages. But overall, it's basically been shown that this is not a very efficient design for identifying the MTD. And if you want to explore this sort of thing, I've included a link here to a very nice app for actually evaluating designs like the 3+3. Okay, so what might you use instead then? Well, you might use what's known as a model-based design. Now I'm going to talk about the CRM, which is a particular model-based approach. So to be technical, it's an adaptive Bayesian design using a parametric dose response model. So essentially what's going on here is that you're actually using a statistical model to estimate the relationship between dose and DLT risk. And then it's this model that advises you on your dose escalation decisions and helps you identify the MTD. So evidently this model is going to become really fundamental and key to this approach. So what is this model? Well, it's basically just a mathematical function that increases in dose. So I mentioned earlier that you're assuming higher doses lead to higher levels of toxicity. So you might see this written, for example, as F beta D. Beta then are just a set of parameters that actually alter the shape of the dose toxicity relationship. And D is just your particular dose. So there's a whole bunch of different models that have been proposed. And there are some comparisons out there in the literature to advise you on the advantages and disadvantages of each of them. I've just included one example here that's known as the one parameter logistic model, which, as you can see, this thing is quite complicated. So it can be a little bit of a daunting task if you're going to use the CRM to actually choose what dose toxicity model to go with. But fortunately, we can ensure that the chosen model actually has a sensible shape over the doses initially by specifying what's known as a skeleton. And the way that you do this is you, usually with clinicians, elicit your sort of set of expected DLT probabilities at each of the dose levels. And then we use this in making sure that this dose toxicity model makes sense. So what about the CRM then? What are its advantages and disadvantages? Well, there are a whole bunch of papers now that show this design is far more likely to actually recommend the correct MTD and dose far more trial patients at close to the MTD. So that's its clear advantage here, really. So what are its disadvantages? Well, it is more complicated. There's no getting away from that. However, I would argue that there is a lot of guidance available now on how you actually use one of these designs. So another figure from this paper I mentioned earlier has mapped out this nice diagram that explains how you would go about actually using the CRM. Another strength here really is that a lot of extensions have been proposed now for this design that make it a bit easier to use in practice. So at least initially, some concerns about the CRM were that it dosed quite a large number of patients at very high doses. However, these extensions allow you to do things to take that into consideration and make sure it doesn't happen. So for example, you can ensure that it only increases dose by one level at a time, or it won't actually allow dose escalation if you have a certain number of patients experiencing a DLT. And I think a really important thing to note here is that ultimately, while a lot of work needs to go into specifying the CRM, once you've done that initial work, actually conducting the trial really isn't any more complicated than using the 3+3. So although there is a model in the background here doing some quite complicated work, the data that you can possibly gather from the trial can be anticipated, and we can use that to put together what's known as a dose transition pathway. So this tells you exactly what doses you would use for each cohort at every stage of the trial. And the way that you read this is you start in the middle of the diagram, so it's got a 1 there, and then there are 4 things that can obviously happen. So if you have a cohort of 3 patients, there can be anywhere between 0 or 3 DLTs. So if we see 0 DLTs, for example, this would say that we then give the next set of 3 patients dose 4. And then if 2 of those experienced a DLT, we de-escalate to dose 2, and so on. And you can build up this diagram so that although there are very complicated mathematical things going on in the background, the actual dosing of patients in the trial is just laid out in a map for you to follow. So that was my quick comparison on 2 types of phase 1 designs. So now I'm just going to speak quickly about some non-randomized phase 2 designs, because these are very, very common in phase 2 cancer studies, in massive contrast to other disease areas where pretty much all phase 2 trials are usually randomized. So in a cancer setting, then the primary outcome in these trials is usually tumor response. So this is just a binary yes/no outcome. So if we denote by P, then the probability of a patient responding to treatment, what these are actually testing is whether or not P is less than or equal to P0, or whether it's bigger than P0. So P0 is some uninteresting response rate, and we're hoping to show that the drug has a higher response rate than this. And we set up our trial typically to actually power the study at level 1 minus beta at some interesting response rate P1. And we make sure that our type 1 error rate is controlled to some small value alpha when the drug doesn't work, so when P is actually equal to P0. So the simplest possible design then you can use in this setting is just a single stage design. So how this would work would be that you'd go ahead and gather data from little n patients, and you'd reject the null hypothesis if bigger than or equal to E patients respond. And the values of E and N then are just determined based on the parameters of your study. So for example, if P0, the uninteresting response rate, is 10%, the interesting response rate P1 is 30%, and we're targeting a 5% type 1 error rate and a 20% type 2 error rate, then what we'd need is 25 patients for our study, and we'd reject our null hypothesis if at least 6 of these responded to treatment. Now, what's nice about these trials is a bit like the dose transition pathway I showed earlier, you can actually draw a diagram that explains everything that can happen in the study. So that's what's shown in this picture here. Along the x-axis, we have the number of patients that we've gathered data from so far, but on the y, we have the number of responses we've seen. So because the number of responses obviously has to be less than or equal to the number of patients, this creates this triangle type shape. And at all points, we're just going to continue our study until we've managed to gather 25 data points. And then if there's at least 6 of these are a response, we reject our null hypothesis. Otherwise, we do not reject if we're down in this bottom right-hand corner. So this is a very, very simple design. So you can ask the question, how can we actually improve on this and make it more efficient? And the way that people have historically done this is to use what's known as Simon's two-stage design. So this includes an interim analysis for futility after some N1 patients, I've called it here. So we'd stop for futility if less than or equal to F of these had actually responded to treatment at that point. So this is really exploiting the fact that in a clinical trial, data is accrued sequentially. So it takes time to recruit these patients. It takes time to collect their data. So we have the possibility to take a look early on in the trial and potentially stop early. And what this is doing then is, because you're looking at the data early on, you have the possibility that you need a smaller number of patients. So on average, if you were to do a large number of these trials, you'd expect the sample size that you required on average to be smaller than that single stage design I had on the previous slide. And you can see the effect of this in this diagram here. So there's an interim analysis after 15 patients where we stop if either 0 or 1 of them have only responded to treatment. And that means that we can no longer be in this bottom right-hand corner where there aren't any little circles. And that's where the sample size saving comes in, because those states are just impossible compared to the previous design. So this is one way that people actually improve efficiency a lot in practice, but there are a whole bunch of other extensions available now that can improve on this further. And I'm just going to quickly go through one of them, which is known as curtailment. So to see why curtailment might be a good idea, you can just consider again this design from the previous slide where we were going to recruit 25 patients and reject our null if at least 6 of them responded. So suppose then that after 23 patients, we'd only seen 3 responses. Well, then there's absolutely no way we can reject our null hypothesis, because we've only got 2 patients left to recruit and we'd need to see at least 3 responses. So that's impossible. So it's been argued then that it's completely illogical to carry on the trial in that sort of scenario and recruit 2 further patients. So this is what curtailment is. It's terminating the trial early as soon as the final decision is confirmed. And this can really bring very large efficiency gains to your study in terms of reducing the required sample size, quite often far further over Simon's two-stage approach. And while unfortunately this hasn't caught on as often in practice as I think it probably should have done, at least formally, it is something that we found quite a lot of evidence of people doing in the literature. So there are a large number of papers that say, you know, at this point we've seen that there was no way we'd reject our null. And so the TSC or the DMC took the decision to terminate the trial. So what does this look like in terms of the diagram I showed on the previous slide? Well, it depends on whether we're allowing curtailment only for futility to say the drug doesn't work, or for futility and efficacy. So if we're going to stop as soon as we are able to reject our null hypothesis, this diagram looks very different because we're actually terminating the trial as soon as we see 6 responses, so potentially as few as after 6 patients. Whereas if we're only stopping when we can say that we will not be able to reject our null hypothesis, we just cut out this bottom corner here. But in either case, again, because we are chopping out the possibility that we end up in a certain number of states, these trials are more efficient over a standard approach. So that's one way then of making a single arm trial more efficient. But I'm just going to conclude now by going through a couple of options for randomized designs in phase 2. So while these have, as I said, historically been used a lot less in phase 2 cancer trials, because of the increase in sample size they require, typically, if you're keeping the error rates constant, a randomized trial will need about 4 times as many patients as a non-randomized study. But recently it's been argued basically that because randomized evidence is just so superior to non-randomized evidence, we need to move more towards using randomized trials in phase 2. So this has led to a large number of proposals in the literature, either for making a randomized trial more efficient so that perhaps they require a smaller number of patients, which makes them closer to a non-randomized evaluation and therefore more feasible, or there's proposals that actually look to make the results from a single arm trial more reliable and more generalizable. So this first diagram here just shows one very simple possible way of making a randomized trial more efficient. which is just to include an interim analysis. So break the study up into 2 stages, potentially stopping after stage 1 in the same way that Simon's 2-stage design improves over that fixed sample design I showed. This approach can improve over a standard single stage RCT. However, there's a limit to how much this can achieve. So though, I'm just going to go through a couple of alternative options instead. So one of these is known as a single to 2-arm randomized trial design, or START design for short. And this is actually the approach that was recommended by a task force of the EORTC, so that's the European Organization for Research and Treatment of Cancer. So they recommended that a single arm assessment precede a randomized assessment within the same trial in order to shorten development time. So that's what this diagram shows here. We start with just a single arm evaluation. And then at that point, if it does look like the drug is doing okay, we then proceed on to a randomized evaluation after an interim analysis. And you're basically arguing there that you're reducing the chance that you move to this randomized evaluation for drugs that are ineffective. So that is more efficient than always just jumping straight into an RCT. Another alternative then, so the previous slide is basically looking at, how can you limit the amount of times you conduct an RCT that doesn't find an effective treatment. These last 2 approaches I'm going to discuss are about making the design a little bit more like a conventional single arm approach. So this first approach I've called a calibration design, although it does have slightly different names in the literature. So how this works is it relies on you having some external data available that you're able to summarize and to describe what you expect to happen in the control arm in the study. So for example, it might be that you think based on historical available data, about 30% of patients will respond to treatment, and you have some uncertainty around that value. So you think it might be as low as 20% or as high as 40%. And then in your first stage of your trial, you recruit a small number of control patients, and then you actually compare these to each other. So you compare your expectation from the historical data to the concurrent control data, and you use that based on how well they match to decide how many controls you need in the second stage of the trial. So you're hoping here that if you got your expectation right, that you then don't need a large number of control arm patients, and you get back to what looks like more like a conventional single arm trial by stage 2. And the final thing I'm just going to mention is what's known as having a synthetic control arm. So this is presupposing that you have some large external data set of results for patients on the current control. And what you're going to do is you're only going to recruit patients to the experimental arm. And then based on the baseline characteristics of these experimental patients, you sample from your historical data set to actually match these to each other, typically using a statistical approach known as a propensity score. So you're basically looking here to make sure that the experimental data that you're concurrently gathering is as similar as possible to the historical data that you've sampled from. And a good example of where this was done recently was blinatumomab, which ended up receiving accelerated approval for a particular type of ALL, that's a type of leukemia. So they conducted just a single arm trial, and they used a very large registry to actually sample from, and showed that the patients that they'd sampled were very similar in their baseline characteristics to the current experimental data. And therefore, this is far more reliable than just doing a conventional single arm trial that compares just to some specified response rate. So I really think this could be a very important approach moving forward for phase 2 trials. However, the big limitation obviously at the moment here is this assumption that you have a very large external control data set from which you can sample. Okay, that was the last of the designs I was going to mention. I've just included a couple of references at the end here on the various things I've talked about.